the very left, there is entry.
Magda, are you there?
Ram, she has some problem with her computer.
Okay.
She said she will reboot and she will rejoin.
Joan will not be joining today. She has some work
We
to
can
do
get started.
in 30 seconds. Okay, hello. Happy New Year to all of you. A warm welcome from the panel back to the ZOOMinar on proteinopathies. Today we have a combination of mentor and mentee from the Oregon State University as speakers, Professor Elisar Barber from Oregon State University, and a graduate student from her lab, Heather Mason Forsyth. Dr. Barber obtained a PhD from Portland State University. After completing postdoctoral research at the University of Minnesota, she joined the Ohio University as an assistant professor in the chemistry department. Then she made a move to join the Oregon State University, where she is currently the head of the Department of Biochemistry and Biophysics. Dr. Barber has received many honors, including the NSF Career Award, Erskine Fellowship from the University of Canterbury, Milton Harris Exceptional Achievement in Basic Research at Oregon, and the inaugural College of Science Impact Award. Her research focuses on the molecular processes governing the transcription factors regulation by IDPs, and now also the coronavirus proteins. The second speaker, mentee, Heather, is a graduate student in the Barber Lab. Heather has a bachelor's degree in biology. Her thesis research includes protein dynamics and biophysical characterization, and very recently she's working on the coronavirus proteins. I'm told that she's also popular with her science TikTok. So with that brief introduction, I would like to welcome both the speakers. Elisar, are you ready to get started?
Yes.
Floor is all yours. Get started.
Okay. Thank you, Rams, for the nice introduction and for the invitation and for this series. Thank the organizers for this. It's a great idea, especially in these times where science is under attack and we have a responsibility to combat anti-science and anti-facts and make science more relevant and more accessible. So with this in mind, we've so our lab is focused on disordered proteins that have or dynamic large complexes that have a lot of disorder and multivalent interactions. And recently we've included work from on SARS-CoV-2, as we will show you. It's an emerging story from the lab. So briefly, disordered proteins function without having to have a structure, contrary to the structured proteins that we all know. So they, some of them fold upon binding, and some of them remain partially disordered, and these are referred to as fuzzy complexes. The class of proteins that we study, which we consider the new class, we refer to as IDP Duplexes. Duplexes because you have disordered proteins that bind a dimeric folded protein with two symmetrical binding grooves to form a multivalent or to form a bivalent assembly where you have a duplex. That's where the duplex comes from. And so NMR has been a powerful tool to characterize these kind of complexes because NMR can map the sites of interactions and can also identify if there are changes in structure or dynamics that accompany binding. And so here we have an example of a protein that folds at the site of binding, but remains disordered in the rest of the protein. And so this is in this cartoon, we show what an IDP duplex scaffold is. So we have two disordered chains that have sites for binding a dimeric protein, but also are prone to self-association, so that binding of one site promotes self-association or promotes binding of another dimer. And if you measure the thermodynamics of these interactions, the bivalent, what we call the bivalent complex, which is already preformed by binding a dimer, binds the green protein with much higher affinity, in this case 50-fold enhancement than the green protein binding the monovalent. And that enhancement in affinities comes from entropy, because the first binding event pays the entropic cost. So then the second event can happen with much higher affinity. And the second event could be also a weak dimer that's becoming a strong dimer, as we show here. Now, this is different from the multivalent assemblies that lead to liquid-liquid phase separation, as shown here in this cartoon. These are multiple weak interactions that can result in higher order aggregates. An IDP duplex is much more specific and results in a duplex or a dimer, two chains coming together. So we asked the questions, how prevalent are these IDP duplexes in biology? And what are multiple ways that they impact function? And today I'm going to tell you three stories, one in transcription factor, one in rabies virus phosphoprotein, and then Heather will be talking about the nucleocapsid phosphoprotein N. And a reminder that the challenge in these large dynamic complexes is that they have extreme level of structural and compositional heterogeneity. And so multiple techniques will have to be used to characterize them. So the common, up to now, the common feature of these IDP duplexes that we study is that they bind a protein that's a dimer, this protein in green, that we call LC8. This protein has been originally identified as a subunit of the dynein motor protein complex. So this is a cartoon of dynein. This is LC8 dimer binding the intermediate chain that is highly disordered. It's a domain of about 300 amino acids that are fully disordered. But binding of LC8 creates a scaffold on which other proteins can bind. Now, this protein, even though was first thought to be only a subunit of dynein, is shown to be ubiquitous everywhere. We see it in the cell cortex, and the nucleus, and the cytoplasm, and the mitotic cells, at the kinetochores, and at the spindle poles. And it's very highly conserved, and it's a dimer with symmetrical binding groups. So while it's been identified as a dynein light chain, we proposed more than 10 years ago that it actually acts as a hub because it binds a lot of different proteins and involved in these complexes without that are not part of dynein. And these are some of these examples of proteins that it binds to. And we've recently created a database where we put all these binding partners in. And all of these are diverse interactions and also have diverse functions. So it's a lot of different processes, and we've studied several of these, but what's common among them, among all these partners, is that they have this same structure. The LC8 dimer binds the partners shown here is a small peptide in yellow, and when it binds the larger protein has high level of disorder, as shown here. The high level of disorder could be then promoted to form a coiled coil, as we see an example with a protein called swallow, or a self-association distant from binding as a protein in the intermediate chain, or a protein from the nuclear pore, NUP159, which has 5 sites for LC8 binding. So LC8 binds at 5 sites, and that's why we call this multivalent. So the same dimeric protein forms this duplex at which 5 of the same LC8 protein bind. Okay, so that's so these are the duplexes. What do they do? And for this, I'm going to look at a couple of examples. One is a transcription factor called ASCIZ. And I'm showing this model here to underscore that characterization of these multivalent, highly disordered proteins requires more than one technique. So we've made use of NMR and electron microscopy. So the function of this IDP duplex is in feedback regulation. So what this means is we have this transcription factor that has a DNA binding domain shown here in red, and a long tail, this long protein, 823 amino acids, long tail of disorder And in this disordered regions, there are motifs for binding LC8. So all of these cylinders here are places where LC8 can bind. So this protein has a potential of binding up to 11 dimeric proteins. So you can imagine that a duplex with 11 LC8s that are decorating the disordered tail. And this tail is also the transcription activation domain. So this ASCIZ protein is a transcription factor for LC8. So it binds the LC8 promoter and produces LC8. And it also can bind LC8 by negative feedback loop. And upon high production of LC8, LC8 binds ASCIZ and stops transcription. So it can recognize the concentration of LC8 and stop transcription when you have a lot of it. So it can sense the transcription level of LC8. And remember that LC8 is a hub protein, so it's involved in a lot of interactions. So you need a protein that can regulate how much LC8 you have in cells. Okay, so we wanted to ask the question of why we have multiple, why we have a duplex and why should be multiple sites for LC8 binding and how does that regulate transcriptional activity. And this is work done from a former graduate student, Sarah Clark, when she was in the lab. So first, ASCIZ is a conserved protein. And more importantly, it has, or the domain architecture is conserved. So you have a DNA binding region, and you have long disordered tails, and it's decorated with potential for LC8 binding. And the number of LC8 sites, the number of these motifs vary with or go down with the length of the disorder. And we look at from two species, from human and Drosophila, and that's because there is cell biology done on these two. And in the biophysical detailed characterization we do with the Drosophila because it's smaller, more tractable. And in all of this work, we're going to look at the disordered domain, which is independently disordered, and the goal here is to look at how it binds LC8 and how many and why is that important. So first, we make peptides of these motifs. So this QT refers to the motif to the LC8 site or the motif at which LC8 binds. And we made 14 amino acid peptide for each of these and tested their binding. Some of them bind very weakly, some of them don't even bind. But when you put them in the context of the full length light chain binding domain or the disordered domain, they bind nicely and they show suggesting positive cooperativity, much tighter than the peptides. And you can even fit them to a simple model and get a stoichiometry of 7, which is what you expect, 7 sites. So if we stop here, we get a model like this. So we have 7 possibilities for LC8 binding. You have 7 sites. So we have kind of a ladder-like structure and nice homogeneous complex. But when we do other experiments, and the ones that I will show are analytical ultracentrifugation or sedimentation velocity, where we follow this by we do titration followed by sedimentation velocity. So here we have a single peak for the light chain binding domain and a single peak for the LC8 dimer. And you start titrating LC8 and you get a complex, but at the saturating concentration, you get multiple complexes. So you really never get a fully occupied complex, and the more stable complex is a combination or is an ensemble of multiple proteins with different occupancies. And so this is, so then you have a dynamic ensemble of complexes that is with a stable low occupancy intermediate. So the intermediate has a bigger peak, so it has more, it's much more stable than the fully bound. And you can identify what these are doing native mass spectrometry, which we did for smaller domains. So you can see there are variation of occupancies in this complex. And you can also see them by looking at negative stain electron microscopy, where it was shown here. So you can, even though the signal to noise is very, very low, you can go pick how many you can actually see how the number in the complex, and by signal averaging, you can get a tumor, 3-mer, 4-mer, etc. And we did this for both the human and the Drosophila, human, which has 11 sites, and we got distribution of all of these complexes, and the ones that have higher numbers, we have many lower population. And so then the model that the apparent from working with both of these proteins is that you have at lower occupancy, you have a stable complex, and then the more you add LCA, the more it can fray out. So you have multiple possibilities for complexes. So why do we? So then it's clear that we have a dynamic ensemble of complexes rather than a one tight complex. So why is that important? So to ask that question, we turned to the human version and made mutants that remove motifs for 3 or 4 at a time. So we can have a set of mutants that can only bind 7 or 4 or 3 or none at all. And we characterized or verified their binding by IgG, and from the stoichiometry we verified that the number that we expect to bind based on the mutations that we made, and then used a transcriptional activity assay using the Luciferase assay, measured the transcriptional activity. And we found, even though the difference is not very, very large, we did find a trend where you have lower motifs, the proteins with the lower motifs have less transcriptional, have more transcriptional activity. So more LCA binding will suppress transcription, less LCA binding will increase transcription. And so the model then that best fits all this data is we have ASCIZ as a complex and dynamic equilibrium with partial occupancy. It doesn't have a lot of LCAs. Or doesn't have a fully bound LCA, but that's the most stable part. But at a higher concentration of LCA, you form this that is not the fully bound, which is not highly populated, only populated at increasing LCA. And when you have this, you stop transcription. And when you have very little LCA, you increase transcription. So rather than having a on and off switch, it's a tuning effect, and you have these dynamic ensemble or dynamic equilibrium of complexes that can sense the concentration of LCA, so they respond to that without making a big increase in transcription, and that's like a buffering effect. So you need both, then multiple. So that's why we do need to have multiple multiple sites for LCA to bind, is to create this complex with that is responsive to concentrations of LCA in the cell. What we didn't answer here is why is it that upon binding LCA you stop transcriptions? So but this reminded here that this is also the transactivation domain. So with LCA binding here, it could compete with co-activators. So that's the story with ASCIZ, which has 11 or 7, depending on the organism, multivalent sites. The story with the rabies virus phosphoprotein is it actually has one site for binding LCA and a dimerization domain. So it's still multivalent, but at the opposite extreme. And this is work done by Nathan Jesperson, also a graduate student. He was a former graduate student in the lab. So the little background on the rabies virus, so it's a protein that it's a virus that has only 5 proteins. And to highlight the phosphoprotein is shown here. It connects the nucleoprotein in green to the polymerase. And that interaction is important for replication, or that's what's required for transcription replication. And so this is looking at the domain architecture for the rabies virus. It's around 300 amino acids. You have a dimerization domain that's studied independently, and they have a C-terminal domain that's also the structure is sold. We don't have a structure for the full-length protein. That's because you have these large, long, disordered linkers. But what's important to us is that in that disordered linker, following the dimerization domain, we have a site for LCA binding. And that LCA is coming from the host. And there has been work that, quite a lot of work in the literature that shows the rabies virus binds LCA, and that in mutant virus that cannot bind LCA, those mice that were infected with the virus live, but those that with the wild type, they 100% fight fatality. So LCA is doing something. There's still infection, but there's no fatality. Now, I'm not saying we have an answer for this, but that is, well, why is this? But that is a good incentive for us to look and see what is LCA doing in this interaction. So we also take a purely biophysical, at least in the beginning, biophysical approach, where I want to remind you this is a dimer of a 300 amino acids. So doing NMR on the full-length protein was a big effort, but we were able to do that. So, but NMR alone was not enough to tell us about the structure. So we have several other techniques, most notably is small angle X-ray scattering and molecular dynamic simulation. And so with this, we have one of the structures. You get multiple structures, but which is what we kind of expect. It's a dimer. We have the structure of the dimer from X-ray crystallography. You have the structure of the C-terminal domain as well, and the rest are disordered. And this we have it from small angle X-ray scattering and molecular dynamics. We get a distribution of these potential structures that are multiple of them because of the linkers separating the folded domains. And so if you overlay these structures, this is the kind of ensemble that you get. Now you do similar analysis with the rabies virus phosphoprotein that's bound to LC8. And LC8 bind to a shorter linker to a 10 amino acid motif. And it doesn't fold the protein, still have a lot of disorder left. And you also get a distribution of structures, but it's a different distribution. And when you overlay these structures on top of each other, you get a different ensemble. And most notably is that in this ensemble, so this is to guide you, the LC8 is the red, the C-terminal domain is the green, and the dimerization domain is yellow here. And what we see immediately is that you have kind of separated the folded domain on the top and the disordered domain at the bottom. And this becomes interesting when we compare it to a sister protein of rabies virus phosphoprotein, and that's called VSV. So VSV protein, this is a domain architecture. It's similar to RAFP, but it doesn't have an LCA site. Instead, it has a longer dimerization domain and a shorter linker. So effectively, what we see by techniques similar to what we've done here, NMR and small angle X-ray scattering and molecular dynamics, is that you have the folded domain at the top and the disordered domains at the bottom. And so what LCA is doing effectively is in increasing the dimerization, is making the dimerization domain longer and restricting the mobility around that linker. And we also tested, rather than just depending on what's been in the literature, we tested to see if indeed LCA has effect on replication. So we used the Minigenome Luciferase assay, which means in using the nucleoprotein, the phosphoprotein, and the polymerase And in this phosphoprotein, you have then a mutation. It just triple ally at the site of LCA. It will inhibit LCA binding. So here we don't have binding. Here we have binding, and we see a big difference in the polymerase activity. So the model that we come up with is that, as shown in this cartoon. So here you have the nucleoprotein that's, and this is the polymerase. And in the beginning, as I told you, the phosphoprotein shown in this cartoon connects the polymerase to the nucleoprotein, and that's what. And this thin thing that's coming out is the RNA. So that's important for RNA. For RNA or for application, and so you need to have this particular shape. While without LCH, you have a multiple or conformation heterogeneity. So this the production of or this polymerase activity is less efficient. So it doesn't have to be a big change. Such a small change can give a big difference, but because you're restricting now the conformation of this duplex. So to summarize what I've told you so far, I've shown two examples where that involve multivalency and dynamic complexes. One is in ASCIZ, where you have so many different multiple LCA sites that forming a duplex. But this duplex is in dynamic equilibrium that can sense better that can be concentration sensor and give gradient of affinity. So you have this multiple LCA sites that gives you gives multivalencies because of multiple LCA sites. And we have an example here where multivalency is only one site, and self-association that also creates a duplex. But in this case, to LCA is during it's changing one from inactive to active by simply restricting the dynamics around that flexible linker. So it does tune and also could tune the dimerization strength. So we have compositional heterogeneity, where you have, in addition to conformational heterogeneity, you also have compositional, means multiple numbers of LCA bound. And here you have conformational heterogeneity. You have one LCA bound, but multiple conformations. So Heather is going to tell you now about a recent story, a very emerging story in the lab about an IDP duplex that doesn't bind LCA, but it's still an IDP duplex, but that we learned to approach it by using the same skills that we've learned to look at IDP duplexes that bind LCA. So I'm going to stop sharing.
Okay. Good morning. So whenever we compare the rabies virus and the SARS-CoV-2 virus, we see some similarities in their protein structures and those functions of those proteins. So in the rabies virus, we have the phosphoprotein that bridges the nucleocapsid protein with the polymerase. But what we see in SARS-CoV-2 is that the nucleocapsid phosphoprotein is actually one protein, serving a similar role of importance for viral RNA packaging and replication and transcription. And when we compare the actual domain structures of the phosphoprotein with the nucleocapsid phosphoprotein of COVID-19, we see a similar layout with two structured domains of either a nucleotide binding domain or the oligomerization domain, separated by small linkers. Looking closer at the SARS-CoV-2 nucleocapsid protein, we know that the C-terminal dimerization domain forms a strong dimer on its own, and the N-terminal domain forms a monomer on its own in solution. And typically, for the most part, these domains have been looked at in isolation, not in the context of their full-length protein, especially with regards to biophysical characterization. But it is thought that the protein exists as a dimer because there's this strong dimerization domain, but there's some conflicting data about what state the protein is in solution. And it's still unclear exactly how the nucleocapsid protein goes from a dimer to a fully formed nucleocapsid inside of the virus with many copies of the N protein protecting and wrapping around the RNA. So we started off with multi-angle light scattering to confirm the dimerization state of the N protein. And what we saw here in purple is that the full-length protein was predominantly a dimer, almost exclusively a dimer in solution with a molecular weight around 100 kilodaltons. And we saw that the C-terminal domain on its own was a dimer, which we expected, and we expected to see the monomer of the N-terminal domain, which we also saw. We confirmed that the full-length protein was a dimer in solution using native mass spec. And using sedimentation velocity, we were able to see that the individual domains gave S values corresponding to a S value that is consistent with either the monomer for the N-terminal domain or a dimer for the C-terminal domain. And you see sharper peaks here because they are predominantly one structured state of their protein. And in the full-length protein, we see a broader peak because of the flexibility that's present in the full-length protein. So to probe this further, we used circular dichroism to observe the secondary structures present in the full-length protein. And most notably, we see this dip around 200 that is reflective of the large amount of disorder that remains in this full-length complex. And when we compare that to the individual domains, we see that the NTD and the CTD are more structured. And whenever we add up the NTD and CTD in yellow here, we see that it roughly corresponds to the full-length protein spectrum, but without the disorder that's present in the linkers that it's missing. So we were able to get a spectrum of the full-length N protein dimer. So this is a 100 kilodalton assembly here. And we see that the protein is a mixture of really well dispersed peaks and really peaks that are overlapping here in the middle that are harder to parse apart. And that's reflective of disorder present in the protein mixed with more structured regions. So we ran a NMR CleanX experiment, and this shows the most disordered regions of a protein. And so whenever we overlay that with the full-length protein, we see that all of the most disordered residues appear within this region. And then we got a spectrum of the N-terminal domain on its own, and we saw that this corresponds to the well-dispersed peaks that are present in our full-length spectrum. So what we were able to conclude is that in the full-length spectrum, we're able to see really clearly the most disordered linkers and the N-terminal domain, but the C-terminal domain remains not visible in the full-length context of NMR, which reflects the model that we think is happening. So at the C-terminal end, it is going to be heavier and tumbling more slowly. Work that's been done previously by the Gladfelter group looked at different pieces of the viral RNA and its propensity to cause liquid-liquid phase separation. And so they found that this 1,000 nucleotide RNA piece at the beginning promotes liquid-liquid phase separation. So for our work, we looked at just this piece in context of the full-length protein. So using mols, we added a small amount of this 1,000 base pair piece of RNA, and we saw a significant shift in size of the full-length protein. So here on the right is what the dimer looks like, and then what we got from adding just a small amount of RNA was a complex that roughly corresponded to about 70 N protein dimers attached to a piece of RNA. And when we compare that to the individual domains, we see that the individual domains at this ratio of RNA don't bind very readily to the RNA, and their primary peak remains in their unbound state. So looking at this closer, we know that the N-terminal domain is the RNA binding domain, and we know that the C-terminal domain has also been shown to bind to RNA in the context of the nucleocapsid. And so what we saw with RNA gel shift assays is that with increasing amounts of protein, you see binding for both the NTD and the CTD as this band shifts up. But you especially see it in the full-length N protein. And then for a control, we have LC8, which is not a nucleotide binding protein, and we don't see any shift here in the RNA, regardless of protein concentration. So we also use sedimentation velocity to look at the RNA itself. And with the RNA alone, we see a single peak of a single species of this flexible RNA structure. And then whenever we add protein to this RNA, we see a huge shift in the magnitude of S values, and we see a really heterogeneous mixture of complexes. So what's happening is that the protein is not binding in all of the same way to the same protein. It's creating a heterogeneous mixture of complexes that are very large. So we saw that the protein, this is just to remind you of what the full-length N spectrum looks like. And when we added RNA to this NMR sample, we see that the peaks that correspond to the N-terminal domain disappear because as it's binding, the tumbling becomes slower and heavier, and the peaks don't appear as readily. But we saw that there was little to no change in this disordered region. So whenever the N proteins are binding to this large piece of RNA, we're seeing that the flexibility is remaining and roughly unchanged, and definitely not directly binding in these conditions. So that brings us to our model of what we think is going on. So in solution, the full-length protein exists as a dimer primarily, but we know that it has a propensity to naturally form some higher level oligomers and is probably in an equilibrium of binding. And when we add this large piece of RNA, the N protein starts to bind along the RNA multivalently. And that brings the N proteins closer together, and with increasing concentration, the C-terminal domains form higher and higher oligomers and start to organize the RNA. Whenever we zoom out, we know that it is a heterogeneous mixture. There's not all one binding at once to a certain way. And over time, and with increased concentrations of N, we expect that the N proteins start to interact with each other and organize in such a way that you eventually end up with your fully formed nucleocapsid. So with that, I would like to thank you for your time, and I'm going to pass it off to Elisar to give you a final summary.
They are an emerging class of IDP multivalent complexes. They, what's common among them is most of them bind the LC8 hub protein, and LC8 has multiple, more than 100 partners. So we have a lot of these IDP duplexes that bind LC8. We have a lot that don't. And the characteristic of them is they display compositional and conformational heterogeneity. And what we've shown that one function is that they form the framework or structural framework for for assembly of mechanized complexes like dynein, nuclear porn, and they act as concentration sensors to regulate transcription. They're involved in many diseases. The IDP duplexes that do not contain, but what we propose, what we think is that the IDP duplexes that do not contain LC8 have less functional diversity. And we showed that we looked at the example of VSV, and so which can be only without LC8 or LC8, like comparison of VSV and rabies. Rabies can have two functions, one with LC8 and one without, while VSV will only have one. And so we propose that it could be that LC8 complexes could have evolved to avoid the phase separation that we see in other multivalent IDP duplexes. And with this, I'm going to acknowledge I'll end by thanking people who contributed to the work. You just saw Heather, current lab member Eugen and Joaquin and Seth also contributed to the COVID project. I mentioned Nathan and Sarah as past graduate students. The COVID project is funded by an NSF eager, and we have a team at OSU working on this. And I want to actually acknowledge also the collaboration with the virology group in France, two groups, the lab of Marc Jamin, where we did the SACS and molecular dynamics simulation, and with Daniel Blondel, where we did the viral transcription. And the electron microscopy was done in collaboration with the Steve Ryko at Portland State. And with this, I'm going to thank you for tuning in this on Saturday, and we'll be happy to answer questions.
Thank you very much to both the speakers, fantastic presentations. So please raise your hands if you would like to directly ask a question, or you can post your question in the Q&A folder. We strongly encourage you to come to the panel and ask questions and involve in the discussion. If you can raise your hand, we can enable that to happen. So there's one. So one question from Christian Griesinger. So I'm going to enable him to join the panel. Christian, go ahead. Christian, you can directly ask the question now.
You want me to answer it? I can see it in the.
No, he's in the panel now. You can ask. You can see him there. You can ask directly.
As you like. I mean, I was just wondering whether there is, if you looked at translational diffusion measurements also of the linker region, because if you have this kind of chain of molecules attached to the RNA, you would expect that translation diffusion goes down dramatically. So I wonder, did you measure that?
We did not measure that, but that is something we could do. We should do. The binding is quite tight. I mean, we see it with moles. We see it on a column. So for it to come off and on is not very likely at these concentrations, at least. Mm. But and it was very clear. Excuse me, that the NTD is the peaks that disappeared, and there's a lot of peaks remained. So obvious answer is that it didn't interact, but we did not measure translation diffusion.
Okay, thanks.
Other questions? Okay, I have one, Elisar. So I know it's because the interest in time, you did not present much of the NMR data. So I wonder, these are very dynamic complexes. So it looks like the dynamics is the key in enabling the function. So were you able to measure the timescale of motion of different segments? It looks like structure is the same, whether you fragment or look at in the wholesome. So what about the dynamics?
So with the N nucleocapsid N, we have not measured dynamics. We do not have assignments. This is, again, this is a new project. We have assignments of the NTD, and that chemical shift did not change, but we have not measured dynamics. But with the rabies phosphoprotein, we did measure dynamics. And what spectra that I did not show, which I could show, is you know, one thing that was surprising is actually when you look at the CT, so there is a long linker separating the dimerization domain from the C-terminal domain. So we could look at this, so we look at the C-terminal domain by itself, and we look at it in context of the full-length protein. And it was surprising to us how similar they were. And that we attribute that to that long linker separating them. So they are behaving, I mean, even though it's in the context of 60 or 70 kilodalton, they are still behaving as, or even 90 when LC8 is bound. They are behaving as independent domains. But yeah, I mean, definitely they are, the chemical shift did not change, but the dynamics did change. We published that, and I don't have it now in front of me. But the other important difference when you look at the spectra is the dimerization domain in both RAV-P, the rabies virus, and in SARS-CoV-2. You don't see those peaks. Those peaks are gone. But you see the peaks of the C-terminal domain, which is after the tail. You know, they have a disordered tail and the domain that has different motion than the rest of the protein, because disorder lets you do that. Did I answer your question?
Yeah. It's very interesting. So it looks like they behave like two independent units, you know, though they're tied with flexible linkers.
Yes. And that's the length of the linker allows that. I'm sure if it's a shorter linker, you wouldn't get that. The longer linker just behaves independently. And so it, yeah.
How do they survive proteolysis and other degradation?
Yeah, I mean, I'm glad you're asking. This is an incredibly tough to do all the NMR on this. So with with both the SARS, the N protein and the RAV-P, we use fresh samples. We never freeze, and we put protease inhibitor. And for every experiment, you have a fresh sample. So 2 or 3 days, and then you have a fresh sample, or even less. And we look at gels before and after, and that hasn't degraded. And so every experiment we showed here, whether it was ITC or moles, we just ran a gel before and after the experiment to make sure that it has survived. So and so both the problem is not just the degradation, the other problem is aggregation. So if you leave it at the certain at that temperature for a long time, it would aggregate.
So when you freeze them, do you expect them to, when you freeze and thaw, do you expect them to be in a different state?
We don't freeze. Yeah.
What happens if you freeze them?
Oh, we
get a different protein when you thaw them?
You know, we see a little bit extra peaks maybe, and you know, that's because we're looking at dynamics, even though the HS2C may look the same, the peaks are a little bit broader, or so we worry about that. So we do not freeze anything that has a mixture of disorder and order. we leave them, we just make them fresh and use them, and we're done. We start a new batch.
Okay, Sumay, you are in the panel. You can ask question to the speaker directly.
Yes, I was just wondering, at the end of the summary, you
Yes.
conclude that LC8 didn't kind of avoid phase separation. Where did
this come from?
Yeah.
Yes. So with the, and we have, it is a tantalizing idea. We have not done the experiment, but what so what LC8 is doing is forming these IDP duplexes that are specific, that you're always going to get a duplex, and phase separation is much less specific, and you have multiple interactions. So it's something that we would like to test, is if in these proteins that can do both, phase separate and also bind LC8, would LC8 compete with phase separation just by forming these specific duplexes. So we don't, it's not a conclusion, it's more of a proposal, or something that we would like to look into. Just because phase separation is made by these non-specific, not as specific and weak interactions, while forming an a duplex with LC8 is more specific.
But the heterogeneity that you generate with LC8 is similar to what we see in the phase separation. Is that your conclusion, right? Although you didn't see the phase separation in actual phase.
Yeah, I mean, the heterogeneity is compositional heterogeneity, not in aggregation. So it's not like we have complexes that could be a duplex or more. You know, the composition means it could be binding 3 or 4 or 5, but it's staying as a duplex, not a higher aggregate. Yeah.
I didn't understand, because in Taylor's presentation at the end of the virus, you did propose that the LC8 actually, although it's mainly dimer, but it kind of has potential to form multimer complex. Is that? But you say
No,
it didn't.
Yes, I did not say that. No.
I see.
It can do multivalent interactions on the same duplex. So you can have a so you can imagine a ladder, and you have multiple rungs when the rungs are LC8, and so you can either have 3 rungs or 7, but you wouldn't have a ladder with like three legs or higher.
I think I get it. Yeah, okay. It's a little different from the phase separation multivalence protein.
Yeah, so yes, exactly. And that's kind of in my introduction, I wanted to make that clear, because when people talk about multivalency, they always think about liquid phase separation. While here, these are more specific interactions that could form a duplex, just like a ladder. That ladder is not getting to be higher aggregates.
And you can actually generate heterogeneity through this dimer.
Yes.
Great. Very nice. Thank you. Very nice story.
By the heterogeneity is by compositional or fluctuation of the linker. So both are important. So you have the linker separating these rungs have a lot of conformation flexibility, and the number of rungs is the compositional heterogeneity.
I got it. Thank you.
Okay.
Go ahead, Christian. Yeah.
If I may ask another question,
Sure.
you showed this progression from this protein attached to the RNA, and then finally was coiling up and so on, and ended up in the virus.
That's Heather's question. Yeah.
So are there additional components necessary, or is this RNA plus the attached protein sufficient to do that?
Heather, do you want to answer this?
Yeah, the short answer is that we don't fully know yet. but most likely there's phosphorylation or dephosphorylation that's necessary. I didn't talk about it, but we know there's a big phosphorylation region within the disordered linker of the N protein. And people are still parsing out like what's naturally phosphorylated or what the roles of those would be. And also N protein has to make some stable interactions with the membrane and envelope proteins that make up the outside of the virus. So the jump from the N protein binding the RNA to like the fully stable virod is still unclear.
But do you know the kinase which phosphorylates the protein?
I don't know off the top of my head.
But I mean, it is known.
It is known, yeah. But I don't know.
So then, in principle, then you just put a membrane, you put the kinase, you put the RNA protein complex, and the virus should form by itself. No?
Maybe.
I mean, it would be kind of a nice synthetic biology experiment that you could do from that.
Christian, one thing missing here is we are looking at a 1,000 nucleotide. We're not looking at
the full RNA.
How big the RNA is, what is it, 29? And even like with this 1,000, we're very happy we were able to do that. I mean, never thought we could make a 1,000 nucleotide and look at it by NMR, right? And do biophysical characterization. So the story is just a very little part of the story. I mean, if we can actually look at the full, or maybe make an RNA that imitates what the full one is, and we could have a packaged one, as you're saying. Yeah. And so what we
it's just in order to find out what's the minimal machinery. I mean, then such a constitution experiment, of course, would be a nice one.
Yeah, but I never thought we can actually look at the 1,000 nucleotides by NMR or even any of this.
I mean, you just see the linker, right? I mean, you probably...
And even that, you're doubting it.
No, I'm not doubting it. No. I mean, it was pretty clear, and it was really amazing how little it was affected.
You know, a lot of people talk about the linkers being the things that bind N, and this was a beautiful way of doing NMR to show that it's not really the linker. I mean, the linker for this RNA, it's not the linker. Yeah.
And we have a similar system where we have also a lot of linker regions, and it's principally one of these classical proline-rich regions binds to SH3, but the linkers which are not affected by the binding also stay more or less identical. Maybe, I mean, at the highest concentration or so, maybe a factor of 2. Yeah. So I think that's if the linker is not involved in the phase separation, then I mean, that seems to be a common
What was something I wasn't expecting at all is the difference in affinity between, I mean, I should have expected because we work on these duplexes, between just the NTD, which has all the binding sites, and the full-length protein. Just by, having the full-length protein that makes a dimer, makes the binding much, much tighter than just looking at the binding domain. Which people miss that because you say, okay, this is the binding domain, so that's what we will be looking at. But actually,
but don't the NTD and the CTD also then make contacts between?
They do, but it's very weak, very weak. On their it's very weak.
But still, I mean, even if it's very weak in a multivalent situation, it could also contribute.
Yeah, but the multivalent is promoted by having the full length where you bring the dimer, you connect them. Yeah.
Okay. Yeah.
Yeah.
Yeah. So your systems are quite challenging for high resolution studies. They're extremely challenging. I really appreciate the way you combine all biophysical tools to come up with a big story. That's great, actually. But I would like to know how much is the contribution from cryo-EM you get on this system? Because this is really highly dynamic. What contribution do you get out of this?
Yes. So I want to show you the motto, point you to the motto, which is take risks, insist, and persist. That's what we do. We don't give up the first time. But we are not doing cryo-EM. It's negative stain.
Oh, you said SACs you're doing, right? Okay.
No, SACs and negative. No, we do negative stain electron microscopy because cryo-EM would be too just too challenging for this disorder. But what we're doing, what I showed here, where we found the negative stain very useful, is when we have this ladder-like structures. So with the rabies and the nucleocapsid, we haven't even attempted. It's just too flexible. But when we have LCA binding on that duplex in multiple places, you can start seeing them on negative stain. But then you see a distribution of them, which is very, very challenging. So you never, that. Like with EM, you want a homogeneous sample, and here we're giving the classical example of what we're looking for heterogeneity. Yeah. So we've been actually trying to develop. So this is in collaboration with Steve Ryko and develop methods that would you don't need beautiful data. But you can just then have an algorithm that can pick them up based on a lot of different computation without having to look to see beautiful data. Because you're never going to get beautiful data on this, the data that cryo-EM people like to see, because it's always going to be heterogeneous. We're going to have heterogeneous, but it's heterogeneity that we care about because that's what gives the diversity and function.
Very interesting. Are there any other questions from the panel? Magda, Bikash?
I do have another question.
Sure, go ahead, yeah.
So when you propose this model of multi-dimers FC8 binding to the binding domain, you propose this LIDAR. It's a parallel structure, two molecules of LC8 binding per molecule, right? The LIDAR you're talking about.
Yeah. Heather, do you want to stop sharing so I can share? Just to show what yeah. Okay.
Yeah, the LIDAR. So have you considered instead of this parallel two molecules binding to several 8 LC8 domain, it can be branched. It means not only two molecules binding to LC8, but you have other molecules like 4 or 5, they all form this network.
Yes. So you're saying instead of branching, I don't know if you're seeing my screen here.
The slides before it.
This one here?
Yeah.
Okay. So we're showing here a duplex, and you're saying it should be
multiplex of the LBD. Multi-molecules.
Why would we have a duplex instead of?
Yeah, exactly. Yes.
Oh, she froze.
Yeah, I didn't get it.
Finding how is an IDP duplex different from multivalent self-association that people talk about. Mm-hmm. So we measured this. So this is based on real data. These numbers here, these are with that. So when we make a protein, so when we have two dimers next to each other, so let's say bind the first dimer, this one in yellow, and then bind the green. This binds much higher affinity. So there is no way that it's going to bind just branching to different ones. So that's the cooperativity really restricts it. So if you already have a duplex because you brought in the first one, the second ones are going to get in just because you have the cooperativity. I think that's the best way to describe it. So one question would be, why not take this rather than binding to this duplex, why don't you bring another duplex together?
That's right.
And we have not seen that, but at least when it's two, like in this case, two next to each other, where binding of one makes the other one much tighter, you have to bind it this way, just because of the cooperativity.
Yeah.
Now, if you have several of them, which is an idea that we're trying to explore now. So let's say you have several of them that you already put the duplex in and you have one that comes off. I do not see a reason why they wouldn't branch. Maybe that's what you're asking, is why is it that should be like this instead of maybe branching to some other? I don't think I have in
Yeah,
mind.
you just imagine another LBD
Yeah.
next to your parallel, and then
And
they can.
it
Yeah.
has to, you know, it has to, so the requirements, which I did not talk about, is that binding of this, so maybe you can go back to the structure. I'm glad we're thinking about this. So here, this is the structure, and you have these symmetrical grooves that are actually grooves deep in a pocket. So you need to have a specific motif that goes in the same way as the other one goes in, because there is cooperativity between one site and the other. Okay. Between across the monomer. So there's cooperativity between two dimers, and there's cooperativity between the monomer and the monomer. And if you have those, then and also that you have another, you know, either so if you have the multivalent cooperativity and the cooperativity across the monomer, then I can see that happening. We have not seen it, but it's something we would like to, we would not rule out. It is actually bridging two different duplexes instead of just one duplex. It will always have to be a duplex because of the dimer character of this, but whether it is possible to bridge two duplexes, I don't see a reason why not. If there are multiple of them, just one will not do that. You see, I cannot see that.
I completely get it. I think your dimer priming the dimer formation and priming this parallel ladder is good theory. It's really convinced me in this case. Yes.
Okay, good.
Yeah.
Okay, let's thank the speakers, Elisar and Heather, for this fantastic presentation. Thank you very much for joining your early morning now. I know it's very difficult to wake up at this hour. Thanks for the presentation, and thanks for the participation from the participants. We'll be continuing every Saturday. I know we are going through a very difficult time. We are definitely going to continue without any breaks. So we are scheduled to continue until the end of June. So no matter what happens in this planet. Thanks for joining us. And now we can be on the informal mode. For example, Heather, you can talk about your TikTok.
Yeah, I want to also thank you guys, and many apologies for being delayed, but I had some technical issues of hearing, had to restart my computer. I really enjoy your story. And Rams, I actually have a follow-up question, which is in one question to you, which is, do you, in regard to your SARS model, is the affinity of the nucleotide binding domain the same for all portions of the RNA segment? Meaning, do you see any variability, like some regions of the RNA of the virus get more binding compared to the others? Because this can kind of, you need to have, obviously, for a nice tight binding in the viral particle, you need to have a strings where you have a bunch of proteins bound, then the linker, so they can be moved around and together. And my second question, which I might have missed, and I apologize for that, how the nucleotide or the N nucleotide binding protein or the N protein recognizes the viral RNA compared to the human RNA? How is that distinguishment? So this is my question on that.
So the RNA binding domain will bind pretty much any RNA. We know that it'll bind non-specific RNA, mouse RNA, human RNA, but it's thought that there is some sort of specificity in the actual context of the virus, but whether or not that's, I think the thought is that it's driven by RNA structures, but we still don't really know. as a scientific community, I think.
Yeah, so we don't know exactly if the NTD is the one that binds the human RNA. You know, we know it does bind. Actually, did we look at? Yeah, we did actually. It does. Yeah, whether they bind the same way, we don't know. We didn't do NMR on that. Whether we will be looking at other pieces. The only piece, I don't know if you missed that, but when Heather talked about it, the only piece we're looking at is the 5' end, which is 1 to 1,000. And there's beautiful work with that was actually presented here in your series from the Gladfelter lab, where she showed that the first 1,000 bind, the last 3,000, the last 3' end also, and somewhere in the middle. They didn't look at any like amino acid specificity, but they did show that it does some cause phase separation, some don't. So these are things we would want to look at, but we haven't looked. So I think other pieces of N should be binding to the other RNAs as you're suggesting, just to make it tight binding. I mean, maybe more compact binding. But
yeah, I would expect to have a variable affinity for the different fragments and structures, so you can have exactly that particular compactness.
Mm-hmm. Yeah, no, that would be something we would love to look at now that we know we can make the RNA.
Yeah.
That's totally new for us. We haven't touched RNA before.
Mm-hmm. Yeah, thank you. I really enjoyed the talk. It's a very nice blend of all techniques together, just to get a very nice mechanistic
I should have
insights.
shown more spectra for RAMs. Sorry. Next time.
No, I'm fine with less spectra.
They are beautiful spectra. I should have shown them for someone who would appreciate.
Yeah.
I'm joking.
A more beautiful spectrum may not add more information. That's the challenging aspect, right? So what other technique in this planet that you want to have to address?
This native mass spec.
What about single molecule?
Sorry?
Single molecule approaches. Can you use them to
get
more insights?
Yeah. Why don't we do single molecule? It's actually easier because you don't have to purify very much. You don't need very much. We have somebody in the department who does that. And how would you use single molecule?
You can narrow down in heterogeneous population to select subset of population.
Mm-hmm. Yeah. What we, I think what we're lacking with the SARS project is somebody who does the virology. With the rabies, we had collaborators who were helping with that, and I sent a student actually to France to do the work. But with SARS, there's a lot of it's more like what we do will still be at the biophysical level, but there's a lot to do, and so yeah.
Very interesting.
Mm-hmm.
Okay.
I have another question.
Go ahead.
It's okay. So,
yeah, sure.
You said the difference between the two viruses is one have LC8, the other doesn't have LC8,
Mm-hmm.
and then you say LC8 is prevent phase separation, and how this benefits SARS, the COVID-19 without LC8, without this prevention of phase separation? Is there something related?
We've seen, it is something we're still thinking about. Like we've seen, so I showed examples of the work with the rabies virus because that's been really characterized at multiple biological levels. But there's also one thing I did not mention is like the Ebola phosphoprotein. The Ebola virus phosphoprotein has similar architecture to the rabies phosphoprotein, and it has an LC8 site. So it's probably doing the same thing that we're proposing to do with the rabies. But then there's measles that has similar architecture but doesn't have LC8, and there are other viruses that we've because we developed this algorithm that can look for LC8 sites. And so there are other viruses that look like they would bind. With the N protein, that's why we got into it in the very beginning. We're saying, what if LC8 binds, and we'll see what it can do. And it doesn't have an LC8 site. And I, played with the idea of one time that maybe if we engineer LC8 site, then it will force N to stay as a dimer, as a duplex, rather than have this higher order aggregation. And maybe it could be one way of targeting. You don't want phase separation, right? So you don't want phase separation for N. So maybe it would be a way of stopping that. So so it is possible that not every duplex will have LC8s, but it may have other things, like with the N, it has a dimer that can make a duplex. And whether it binds other things, we don't know. I mean, there are all these long linkers that we don't know what they bind. It could bind something else that looks like LC8, or maybe it wouldn't. And putting LC8 site in there may be a good way to regulate phase separation, but it's not what the cells do. It will be more of an addition. I don't know if I answered your question.
Yeah, you answered. I'm actually aiming at comparing different viruses with LC8 or not. And you did answer that.
Yeah. Yeah, no, we have viruses that look very much like the VSV and the rabies virus phosphoprotein. They look exactly the same. And the only difference is VSV has a shorter linker. And so it doesn't need LC8 because it doesn't have that much variability. While the RAFP has a long linker, it needs LC8, so it kind of puts it in, it restricts its motion. But why is that important? We don't know. I mean, like, why? Is there another function for RAFP that without LC8, or that you need it without and with? So it's just adding diversity of function when you have LC8 in there, because it just regulates. Has another way to regulate it.
Yeah, it seems the LC8 is just in the toolbox. Use it or not is up to you. Feels like it's
the cells are using it. We're with the SARS-CoV-2. It's a toolbox, but there are more than 100 proteins that use LC8, and without LC8, the cells die. So we know it's an essential protein. A lot of proteins need it. We're trying to understand what for.
Thank you.
Mm-hmm.
We are not recording it, so this is informal, so you feel free to talk anything that you like to.
A couple of questions, may I?
Yes, sure.
Yeah, my first question is for Elisar. So I think like when they bind, and you mentioned that like the linker is there and two domains are binding. So I would assume that like in the solution you have a more ordered structure when that happens. So do you think that if you will take that sample, that can be aligned in the magnetic field?
So when so I showed these. I don't know. I drew them aligned. That's based on thermodynamics, not from NMR. The reason I draw, I think you're referring to like a disordered protein, and then you bind the dimer, and then you have these two nicely.
You mentioned about the ladder and like so like
yes,
an impression that you have a very ordered structure in your solution.
Yeah. And and again, also this is purely based on thermodynamics. So what we've done is, what we've shown is that when the second binding event happens, you have a lot of enhancement, but that enhancement is all coming from entropy, not from delta H. It's all the delta S, and it showed that there's really no change. That what you have is still disordered, but it's restricted. So it's not you don't have a beta strand that's formed or anything. So so then they are ordered. But I don't think we were able to see any differences in NMR dynamics by measuring like backbone dynamics. We didn't really try more than that.
I have a next question for Heather. So I think in one of your CD spectra, I could see that there are some changes in like when you titrate, you could see some changes in
Okay, it looks like we are done actually. Thank you all very much for joining us, and we can say bye-bye for now. See you all. Thanks, Heather. Thanks, Elisar. Thanks, Christian. Magda.
Thank you, guys.
Where are